Hello guys, welcome to Bio class. So today we're going to discuss about the new chapter that is a sexual reproduction in flowering plants, right? So in this chapter we're going to discuss about structure of the flowers, parts of the flowers. We're going to discussing about the male gamete that is a stamen, or we also calling as microsporophyll. Then we'll discuss about carpel. It is a female gametophyte. We are also calling as a megasporophyll. Then we'll discuss about pollinations and its types. After that, we'll discuss about the fertilization and post-fertilization. So these are the things we're going to study in these chapters. Okay. We'll move to the first concept, that is a flower. Yes, as we know, flower is a reproductive part of angiosperms. So flower means it is a modified shoot with highly condensed internodes. So flower means it is nothing but modified shoot with highly condensed internodes, and this flower is meant for reproduction. Okay, now we'll focus on the parts of the flowers, right? So it is made up of four parts. Calyx, corolla are called non-essential or accessory, right? So here the calyx and corolla are called non-essential or accessory, means so without this part like calyx and corolla, so the flower can be go for the reproduction. It is not mandated that it is a must and should require for the flowers. These are just supporting for the reproduction. So that's why we are calling as a non-essential, or we are calling as accessory orals. Okay. So next one is, there is a androecium and gynoecium are essential orals or necessary orals. Yes, why it is an essential means because without these two parts the flower can't go for the reproduction. So because of that, so these two parts are called essential orals or necessary orals. Yes, we know that in flower, so there are two parts. It is a essential part. One is it is androecium and one is it is a gynoecium. So androecium is made up of stamens. So androecium is, it is a made up of stamens, and it represents the male part of the flower. It represents male part or male reproductive structure of the flower. Gynoecium, it is made up of carpels and it represents female part of the flowers. Okay. So flowering in plants. So we know that parts of the flowers. Now we'll focus on the when the flowers will get blooms. The flowering in plants is controlled by environmental factors like temperature and length of day. So that is what calling as photoperiodism. So this photoperiodism we will discuss in the first year chapter. Means it is a depends on the intensity of the light or how long the plant will expose it to the sunlight. After that it will produce a flower. That is what calling as photoperiodisms, right? So this temperature and length of the day, which exports hormone substances, bring out conversion of from vegetative buds into flowers. So here the temperature and duration of light is important, and these are stimulate the hormones and it will convert the vegetative buds into flowers. In this slide we focus on the structure of the flowers. Okay, so here we can find the calyx, corolla, stamens, carpels, right? So the green part is spreaded out So that part we are calling as a calyx. Each unit of calyx we are calling as a sepals. Below that there is small cup-like structure that is we are calling as receptacle. So further below that the elongated structure that is we are calling as a alamus. Yes, now. So we can find here some colored part. So this colored part we are calling as a corolla. So each unit of corolla we are calling as petals. So these two, calyx and corolla, so both they are the non-essential part. Okay. Next one is we can find here some elongated structure. So this elongated structure they are consist of small lobes on their tips, and this structure we are calling as a stamens. Stamens are represents the male reproductive structure. So in between these two we can find one structure that is we are calling as a carpel, or we also calling as pistil. It represents the female reproductive structure. In this slide, we will discuss about the male reproductive part, that is the androecium. Right. So androecium, it consists of stamens. Stamens we also call it as microspore. Right. So the diagram is here. So we can observe in diagrams. Right. So this stamens, it is made up of three parts. The stamens is made up of three parts. Right. So like filament, anther, and connective tissue. Okay. So observe in diagrams. So there is a small elongated structure. So that elongated slender like structure we are calling as a filament. So this filament at its tips, it consists of a lobed structure that is we are calling as a anther. So we can observe here yellow color part. So this part we are calling as a anther. So this anther, generally it is a two lobes. Generally it is consist of two lobe, or it consists of two sac-like structure. Okay. So because of that we are calling as a dithecous anther. So dithecous anther means the anther which consists of two lobes, that is we are calling as dithecous anther. If it is consist of one lobe we are calling as monothecous. Okay. So we know that filament, what is a filament, and we know that so anther. So next one is it is a connective tissue. So name itself replicates that. So it will connect between the filament and anther, and it will again connect between the two anther lobes. Right. So this is a connective tissues. Okay. So as we know that it is a dithecous. So each lobe consists of two pollen sacs. Each lobe consists of two pollen sacs, or it consists of two cavities. So here there are two pollen lobes. So totally how many cavities are there? So there are four cavities, or four chambers are there. So inside these chambers we can find the pollen grain. So actually the pollen grains will be get produced in this pollen chambers. So this pollen chamber we are also calling as a microsporangium. In this slide we are going to discussing about the structure of young anther. As we know, so anther is a dithecous. It consists of how many pollen sac or pollen chambers? It is consist of four pollen sac, or four microsporangium, or pollen chambers. Okay. So these pollen chambers are made up of four layers. These pollen chamber or pollen sacs are made up of four layers. Okay. So we can observe diagram A and B. Right. So first layer it is we are calling as epidermis. After that it is a endothecium. Then middle layer and tapetum. So these are the four layers. It is found in the microsporangium or pollen sacs. Okay, now focus one by one. Okay, so outermost layer we are calling as epidermis. Outermost layer we are calling as a epidermis, right? So it is a single layer. Here cells are compactly arranged. So outermost layer is epidermis. It is a single layer, and here the cells are compactly arranged, and it is meant for protection of microsporangium. Now what is the use of this epidermis? It is meant for protection of microsporangium. Okay, so you can observe in diagrams, right? So outermost layer we can find here, it is a epidermis. Okay, after epidermis we will find one more layer, that is called as endothecium. After the epidermis we find endothecium. So here the cells are radially elongated and they shows fibrous bonds. Cells are radially elongated and they will shows fibrous bond. These are hygroscopic in nature. The cells are hygroscopic in nature, and this endothecium layer, it helps in the dehiscence of anther at maturity. So when the grains are produced, when they become mature, after that, so it will, by the help of dehiscence process, the pollen will be comes out of the microsporangium. That time this endothecium helps in for coming out of that pollen chambers. That is what we call as dehiscence. Okay. So next layer, that is what we call as middle layer. So middle layer, it is found below the endothecium. It's found below the endothecium, and it consists of 2-3 layers of cells. So it is made up of 2-3 layers of cells. Here it acts as a storage of starch. So middle layer cells are, that helps in the storage of starch, and also it helps in the dehiscence of pollen grains. So dehiscence means, so the pollen grains comes out of that pollen chamber. That process we are calling as a dehiscence, right? So mainly the middle layer, it helps in the storage of starch, and it also helps in dehiscence of pollen grains. Okay, so last one, that is a tapetum. So this tapetum, it is an innermost layer of anther wall. So it is an innermost layer, right? So it is completely surrounding to the sporogenous tissues. So tapetum, it is completely covered to the sporogenous tissues, and this tapetum, it helps in developing the pollen grains. Tapetum layer, it helps in the developing of the pollen grains, and it provides the nutrition to pollen grains. It provides nutrition to pollen grains. In this slide, we are going to be discussing about the microsporogenesis. So we look at it first, its flow chart, right? So by flow chart, we can easily understand the microsporogenesis process. Yes. So microsporogenesis means it is a formation of spores. Okay. So we will now focus on this flow chart. So anther, as you know, in young anther, so it consists of epidermis, and this epidermis are surrounded by parenchymatous cells. Epidermis there, and this epidermis are covered by parenchymatous cells. So they later, these parenchymatous cells are become the archisporial cells, or they are become archisporium. So this archisporium, later it divides, and it will form the outer primary parietal cells and inner primary sporogenous cells. Okay, I am repeating again. So epidermis here, epidermis covered by parenchymatous cells, and these parenchymatous cells will gives to archisporial cells or archisporium. So later this archisporium cells divides and give the outer primary parietal cells and inner primary sporogenous tissues. Inner primary sporogenous tissue. So later these parietal tissues, they are get differentiated into the layers like endothecium, middle layer, and it will form the tapetum. So now this inner primary sporogenous tissues, it will go for again division, it will form the pollen mother cells. It will form pollen mother cells, So after that, it's undergo meiosis process and it will form the pollen tetrad, and finally we'll get the pollen grains, right? So this is all about the microsporogenesis. Okay, it is shown in the flow chart for easy understanding. Okay. In last slide, we discussed about the formation of layers like endothelium, middle layer, and tapetum. After that, we know that how the sporogenous tissue are formed by the help of archisporial cells. Okay, so in this slide, we are going to only focusing on the sporogenous tissues, right? So initially, when the sporogenous tissue are formed, initially the tissue are in polygonal shape and the all cells are closely packed. Yes, when the tissue is formed, that time these are polygonal shape and they are closely packed. Later on, the pollen sac increases, means the pollen chamber size will be goes on increasing. That time the sporogenous tissue become rounded and loosely arranged. So initially it is a polygonal shape and closely packed. When the chamber size increases, that time these are become rounded and loosely arranged. Okay, so these sporogenous tissues, it may divide and form the microspore mother cells, nothing but microsporocytes. So again here the spore tissue goes on dividing and form the microspore mother cells, or we are also calling as a microsporocytes, and we are also calling as PMC, pollen mother cells, right? So there are number of pollen mother cells will be formed, right? There are number of pollen mothers will be formed, but all are not get activated. Their all cells are not activated. Some of pollen mother cells are get degenerated. Yes, they are get degenerated, and these are helps in the providing nutrition to the developing pollen grain cells. Okay, so later the spore mother cells undergo meiosis. So meiosis means it is a reductional division where the diploid cells are become haploid in numbers and produces haploid spores, that is what we are calling as tetrads or pollen tetrads, right? So in microsporogenous tissue, some cells are, what happens, they are become degenerated, and degenerated cells are, they are helps in the developing the pollen tetrad, nothing but it is providing the nutrition, other things to the developing pollen grains or pollen tetrads. Okay, so after formation of pollen tetrads, then there will be the cytokinesis. We know that the movement of cytoplasm, it results into separation of 4 haploid nuclei. So cytokinesis results into separation of 4 haploid nuclei into 4 separate cells. Okay, so after the meiosis, what happens, there are 4 haploid cells will be formed, and due to the cytokinesis these 4 haploid cells are get separated into 4 separate nuclei, and which are arranged in the tetrad. They are arranged in the tetrad. Yes, so we can observe in these diagrams. So initially we are shown here the young anther, it start developing. So here some part show it showing the its layers, endothelium, epidermis, apetum, and sporogenous tissue. This sporogenous tissue undergo dividing and will form the pollen mother cells. After these pollen mother cells or these microsporocytes undergoing meiosis and produces the haploid nuclei. Because of cytokinesis, these 4 haploid nuclei are get separated to 4 separate cells. They are get separated to 4 separate cells, and these separated cells we are calling as a tetrad. This is arranged in the form of tetrad. So here we can observe here one in last diagram. So here observe there are 3 nuclei, means actually one more nuclei is there, it is not appearing here. So this kind of arrangement we are calling as a tetrad. Okay. So in last slide we have discussed about the microsporogenesis process. Yes, so we know that, so microsporogenesis means it is a formation of spores, right? So sporogenous tissues, right? So sporogenous tissues, it goes on further dividing and it form the pollen mother cells. So there are numbers of pollen mother cells will be formed, but all are they are not active. Some of get degenerated and it provides the nourishment, or it provides the food for the further developing pollen grains, right? So this pollen mother cells will goes for the meiotic divisions and produces 4 nuclei cells, or it produces the 4 haploid cells. Okay, so these 4 haploid cells later, by the help of cytokinesis, the all 4 nuclei are get separated to 4 separate cells. Okay, and these separated cells we are calling as a pollen tetrads. We are calling as pollen tetrad, or we are calling as tetrad. So why you are calling as tetrad means here the 4 nuclei are present. So 3 are they are in the interconnected and 1 is present in the center. So it is not visible. So that structure we are calling as a pollen tetrad. Okay. So now in this slide we will focus on one more thing, that is a pollen kit. Okay, so pollen kit, nothing but it is made up of lipids and carotenoids. It's formed by the tapetal layer. Now what is use of this pollen kit means it will attract the insects. It will attract the insects and it will give protection from UV to the pollen. Okay, so here we mention homework. So first one is it is a draw a neat label diagram of typical flower. Second one is it is a mention the functions of calyx and corolla. Third one, draw a neat label diagram of anganther. Yes, so this diagram is important. So we have to draw for 3 times. How many times? 3 times you have to draw this structure. So if you not, the diagram is not clear in this slide, so better you go for the refer the books. From that you have to copy that structure 3 times. Okay, next question is anther is made up of how many layers? Okay, anther is made up of how many layers, and mention the functions of each layer. You have to mention the numbers of layers. After that we have to write the functions of the each layer, and last one it is a pollen tetrad. Okay, so this is all about this first session in chapter 2, that is a sexual reproduction in flowering plants. Okay, further we will discuss in next session. Yes, thank you.